This is part 2 of chapter 8, where we're talking about energy concepts. And we can't talk about energy in a biological context without at least having a brief discussion about ATP. Um, so ATP, of course, stands for adenosine triphosphate. You can see the structure here on the right. It contains a 5-carbon sugar, ribose, at the center, has a modified adenine nucleotide, and then the triphosphate refers to these 3 phosphate groups that are all bonded together. And you can think of ATP as basically the cell's favorite energy currency. So all cellular processes have been optimized to basically use ATP as a quick, uh, easy to burn source of energy. So ATP is really powering uh, all the energy needs of living cells. And as we're going to talk about in the next chapter, some of these other energy containing molecules, bigger molecules, things like sugars and fats and proteins, if cells have access to those molecules, they're going to extract some of the energy from those larger molecules and basically convert it into ATP because that's what it can use most easily and quickly. So, what's so special about ATP? And really, it comes down to these phosphate groups. So, if you look at these, you'll notice these phosphate groups are negatively charged, and you have 3 of them all bound together. And as I'm sure you're aware, like charges do not like to be close to each other, right? Opposite charges attract, similar charges repel each other. So in this instance, you have these negative phosphate groups that are all being forced together, held together, and they don't want to be there. So you can think of that as kind of like a coiled spring. And what that means is you're going to have very high energy bonds here between the phosphate groups holding those together. So the way we extract energy from ATP is basically by breaking one of those bonds between the phosphates, releasing one of the phosphate groups, and hence some of the energy that's stored in that bond. And you're going to see this happening over and over again as we go through some subsequent chapters. You'll see ATP being converted into ADP, diphosphate, because ATP was hydrolyzed. We broke one of these bonds, removed one of the phosphate groups, and we go from triphosphate to diphosphate. And you could even go all the way down to monophosphate, AMP, and that could move in the reverse direction as well, right? We could start with a monophosphate, add a second phosphate to produce diphosphate, add a third to produce ATP. But bottom line takeaway here is ATP is that preferred energy molecule in the cell, and that energy is stored in those bonds between the phosphate groups, and it can be released by breaking one or more of those bonds. And another common theme here you'll see with ATP, and I was just alluding to this, is ATP can be hydrolyzed, so we can remove a phosphate group on ATP and convert it into ADP plus the inorganic phosphate. There's the phosphate that was removed. That's releasing some of that energy that was stored in that molecule, and that can be used to power energy requiring endergonic reactions in the cell. So movements, really, anything in the cell that requires energy, we could use that as the source to power it. And then on the other side of that coin here, we can take ADP, and if there is some available energy in the cell, usually that's going to be coming from some exergonic energy releasing reaction. We can trap some of that energy back into ATP by basically adding a phosphate to ADP to regenerate ATP. So it's very cyclical, right? We hydrolyze ATP, we're left with ADP, energy is released. We can use any available energy to add that phosphate back on and store it back up in ATP. And here's a little bit more detailed view of that same idea. Here's a molecule of ATP being hydrolyzed, so we break off one of the phosphate groups. This is an exergonic energy releasing reaction. It's got a negative delta G to take it back to some of those energy concepts in part one. And some of that energy can be used to power an endergonic energy requiring reaction that has a positive change in free energy, a positive delta G. And the specifics here aren't important at the moment, but this particular reaction we're going to see again in Chapter 9. It's part of the cellular respiration pathway of reactions. But this again is very common. We see ATP being used to power these energy requiring reactions all the time in living cells. All right, so we got some basics on ATP under our belt. Now we want to turn to enzymes and talk about, well, what are enzymes? How are they involved with cellular energetics? And how are enzymes regulated? So enzymes are catalysts, meaning they speed up the rate of chemical reactions. Enzymes are almost always proteins. There are some enzymes that are RNA molecules, but the vast majority are proteins. And the way enzymes work is by reducing what's called the activation energy, or E sub A, that's required to start essentially every chemical reaction. So even a reaction that releases energy like this one, so if we look at this graph here, the products of this reaction have less free energy than the reactants, so we're getting this negative change in free energy. Energy would be released by this particular reaction. Even for a reaction like that, we have this input of energy that's required to get the reaction started, and that is the activation energy. And so what we're going to see with enzymes is the way enzymes work is by reducing that activation energy requirement. So it's going to allow these reactions to happen more quickly. So how can we reduce activation energy? Well, we'll look at a few different mechanisms by which this could happen. So theoretically, this could come from heat. You could use heat to speed up molecular movement. In a lot of cases, that might reduce the activation energy needed to start a reaction. But in biological systems, that's not really very tenable, right? We don't want our cells to have to heat up every time we want a particular reaction to occur, and then they cool back down, and then they heat back up again. We want to maintain a pretty steady internal temperature. So heat's kind of off the table. We talked earlier in this chapter about how heat is not a very usable form of energy in biological systems. So usually what enzymes and ribozymes is the term for those RNA molecules that act as enzymes. What these molecules are doing is usually bringing reactants together in some physical way that's going to help reduce that activation energy that's required. And I'll show you some examples here in a minute. You've probably seen a cartoon like this before if you've ever studied enzymes in a previous biology class. But I want to go through this again quickly. So here's our enzyme, our protein, and every enzyme has what's called an active site, which is the business end of the enzyme where the particular reaction is going to be catalyzed. What happens at the active site is the enzyme is going to bind to its substrate or substrates, depending on the particular reaction. The substrate or substrates are the molecule or molecules that enzyme is acting upon. So in this particular example, this enzyme binds to two substrates, right, and it's going to hold them together in a position that's going to help facilitate the formation of a bond between those substrates. And then once that reaction has been catalyzed, what's now called the product, the end result of that reaction, is released. And as you can see here, the enzyme is basically unchanged. So it looks exactly like it did before the reaction started. And it is in fact ready to catalyze that same reaction again. So at this point, if there's more substrate available, it will bind, catalyze the reaction again, and basically keep going until it runs out of substrate. You'll see this little green molecule down here, kind of lurking in the shadows. It is being identified as a non-substrate, and this is just trying to show you that enzymes are very specific. So, this little green molecule is small enough that, theoretically it could fit inside that active site, but it doesn't have the correct shape to want to actually bind there. So, yeah, maybe it bumps into the active site, but there's no recognition or interaction, so it just would fall out. Whereas the substrates have the correct shape and maybe the correct charges to actually temporarily stick and bind to that active site. And just to take this back to some of our discussions in earlier chapters, the shape of the enzyme and the shape of the substrates is really critical for this reaction to happen. So, if we were to change the shape of the enzyme in some way, change the shape of that active site, it would no longer be capable of binding to these substrates. If you wanted to think of this in terms of an equation, which I know you do, we could write this out as E plus S. So our enzyme is going to interact with our substrate. We will get this temporary enzyme-substrate complex being formed. So that's while the enzyme and the substrate are actually bound to each other. Then the product P will be released. And as I said on the previous slide, we're back to having our enzyme back ready to catalyze the same reaction again. So enzyme binds to substrate, forms this temporary enzyme-substrate complex. Reaction's catalyzed, product is released, and we're back to the start. And just taking this back to this idea of activation energy, here we can see in this graph the blue line representing the energetic requirements for a reaction that is not enzyme catalyzed, whereas the red line represents the same reaction that is being catalyzed by an enzyme. And overall, the energy of the reaction is unchanged. So the delta G for that particular reaction is exactly the same, right? The change from reactants to products is the same in both cases. But the big difference here is the amount of activation energy required to get that reaction started is less if the enzyme is present. Okay, so how do enzymes work? How are they catalyzing these reactions? And generally speaking, most enzymes are doing one of two things. Either they are going to bind to two substrates and help form a bond between those two substrates, or an enzyme may bind to one substrate and often help break a bond, so break that one substrate into two different products. Those are probably the most common types of reactions: form a bond or break a bond. And so one way the enzymes could work is by orienting the substrates just to help facilitate the formation of a bond. So here in this case, again, the enzyme is binding to these two substrates, and it's just going to position them just perfectly so that a bond is going to be very easily formed between those two substrates. And taking this back to this idea of activation energy, if this enzyme was not present, we could still get a bond being formed between these two substrates, right? If they're just floating around, bumping into each other. But it's going to happen a lot more slowly because those two substrates are going to have to bump into each other in just the right position to allow that bond to form. And that's going to take a lot longer than if we have this enzyme there that can say, here, I'm going to grab you, I'm going to grab you, and I'm going to hold you in this position just right so that we can make this new bond. So orientation is a very common mechanism by which enzymes could do their thing. If the job of the enzyme is to break a bond, a lot of enzymes might do that by actually inducing some physical strain on the substrate. So here our enzyme is bound to one substrate, and it's going to try to break this into two pieces. And you can see here, once the substrate binds the enzyme, right, it's actually applying some pressure onto that substrate, and that may help break a bond inside that substrate, and it's catalyzed that reaction. Another common mechanism by which enzymes could work is by altering the chemical charge of a substrate, so adding or removing charges. That's very common as well. And you might have seen these terms if you've ever studied enzymes before, but a lot of times you'll hear it referred to as this lock and key model, and this is kind of what I was talking about a minute ago, where the enzyme and the substrate have to fit together just perfectly like a lock and key in order for the enzyme to be able to work properly. And kind of the basis of that interaction is typically that the enzyme doesn't really change at all, right? It just stays static. These substrates come in and bind to the active site. The substrates get changed, but the enzyme does not. But there is an exception to that called induced fit, and there's a lot of enzymes that work in this way as well. But induced fit is where an enzyme will temporarily change its shape while it's bound to the substrate. But then once it's done with the substrate and releases product, the enzyme goes back to its original shape. So it's just a temporary shape change. Here's an example of that. And here's our enzyme. Here's the active site, this little groove on the left side of the enzyme. And what this is trying to show you is that when these two substrates bind to the active site, the enzyme is physically clamping down. So that's what these arrows are showing. So it's constricting a little bit. The enzyme is temporarily changing its shape. But again, once it's catalyzed the formation of a bond between these two substrates and releases the product, the enzyme will go back to its original shape. So shifting over to this kind of idea of enzyme regulation, I wanted to point out that enzymes do sometimes require some help to do their job. And we're not going to go into a lot of detail here on these particular types of helpers, but I did just want you to be aware that they exist. Prosthetic groups, cofactors, coenzymes, these are all different types of molecules that can, again, help an enzyme carry out their job. And you can see they're all a little bit different from each other. So prosthetic groups, by definition, are non-amino acid groups that will bind to enzymes and help them catalyze whatever reaction they're trying to carry out. Cofactors are always inorganic, meaning they don't contain carbon. They're always ions, meaning they have some charge. And then there's molecules called coenzymes. So these are organic, carbon-containing molecules, and they always bind to enzymes just temporarily. But again, just these three different types of enzyme helpers, if you want to think of them in that context. All right. So I did also, while we're talking about enzymes, just quickly want to show you kind of a typical graph of an enzymatic reaction here, where we're looking at the rate of a reaction as it relates to the amount or the concentration of a substrate that's available. And what we would see if there's no enzyme present is usually just a very slow linear increase. So as we increase the amount of substrate, the rate of the reaction just slowly creeps up. And you'll notice if an enzyme is present, that line looks pretty different because the enzyme is speeding things up so much that the slope is going to be much, much steeper, right? So if we give it just a little bit more substrate, the rate of that reaction jumps way up. And another hallmark of enzymatic reactions is they're always going to reach some maximum rate. And this would basically be where you're swamping out the enzyme. So you've given it so much substrate up here at this point that it just can't work any faster, right? So it's running as fast as it can. It doesn't matter. You can just keep feeding it substrate, and the rate of the reaction is not going to increase at that point unless you bring in more enzyme, more help. All right, I am going to end part 2 right there, and you can pick this up in part 3.